In this last lecture, I'd like to spend some time talking about the applications of transition metals. Uh, and it's just broad. It's so broad. We could actually have several classes on this topic alone, and they would be really fun classes. Um, so, in general, where you see transition metal chemistry come up is in when we're talking about extracting metals from ores. That becomes extremely important. We also use transition metals and what we learn about ligands and coordination compounds to understand how to create chelating agents to help relieve the consequences or symptoms of heavy metal poisoning. We use them in the quantitative analysis of ions, much like we used in our qualitative analysis of ions at the beginning of the quarter. They're used as commercial coloring agents. They're gorgeous, right? I mean, Prussian blue is a mixture of hexacyano iron 2 and then iron 3. And just look at any Bob Ross painting and you will see lots of transition metals in the pigments that make his masterpieces. We also see them as drugs and therapeutic agents. Platinum is used as a chemotherapy agent as cisplatin with ammonium and chlorine. And, and, and one of the really rich areas of transition metals is in catalysis. And transition metals are so tunable that we can design these transition metal coordination complexes to mimic the chemistry of things we see out in nature in different organisms. But we can also, aside from taking inspiration from nature, design small molecule catalysts that involve these transition metals that can help perform the different reactions that we're so interested in general. Anything from converting methane into methanol so it's easier to transport, to cracking hydrocarbons to make it easier to process them into different feedstocks for different chemical processes. And the list goes on and on. Um, it's a fascinating field. And then there's also transition metals in biomolecules. And the body utilizes transition metals to catalyze a lot of the reactions that need to happen in a lot of the processes because again, they're super tunable. And so a protein or a metalloenzyme's environment can make a transition metal do an extremely specific task. And so we see these all over our bodies and all over nature. We see them embedded in porphyrin rings and cytochrome C and hemoglobin and chlorophyll. And we'll talk a little bit about that there. And so at the end of this chapter, I'd like to dive into some of these applications, but there's not enough time to talk about all of this. It's all fascinating stuff. And while I would love to do our last lab, it's just an art project where I send you a bunch of like oil-based paints that all have transition metal colors in it, and I want you to paint your feelings about chemistry. I haven't written that lab, and I don't have the paints to send you, but I know a lot about and care a lot about both catalysis and biomolecules. And so for the focus of applications, I'm going to give a short abbreviated lecture about some interesting points around bioinorganic chemistry as it applies to transition metals. And so this is a big field too. This is a whole class. This is several whole classes. Again, just like transition metals themselves are several classes. And we can really go deep into the chemistry here. In general, though, taking a step back, bioinorganic chemistry is the study of biochemical functions of inorganic elements. And so most of your body is water, but then beyond that, most of your body is carbon and oxygen and hydrogen and sulfur and phosphorus, these organic compounds, which is part of why organic chemistry is so interesting. And the incorporation of metals into our body are not happening on the same scale as carbon, but they play these extremely important roles because of their ability to have multiple oxidation states and to undergo really interesting redox chemistry. And so when we study metals in biology, we're really studying how nature has utilized them to perform different chemical transformations. And we can learn from those systems that have been optimized by evolution and take advantage of them. And so there's definitely trying to learn from biology and how these transition metals can help in the collection, conversion, and storage of energy, which is what our bodies are doing all the time. And then also looking at how catalysis can happen under moderate conditions, like, you know, ambient temperature and pressure, and with water and oxygen around. And a lot of the really interesting catalysis that we do with these transition metals have kind of these catches to it. Like it needs to be under high pressure or at high temperatures, or there cannot be a stitch of oxygen around, things like that. Also, our body utilizes these transition metals and biomolecules to do stereoselective synthesis. Blah, that's a mouthful, but what it means is it means synthesizing compounds like amino acids that are themselves chiral, and doing it so that so remember, chiral means like my hands, they're mirror images, but they're not superimposable. So if you're just sticking atoms together, it'd be really hard not to make about 50% of my right hand and 50% of my left hand. But we can utilize transition metals, and your body and nature utilizes them to make only right-handed molecules. So chiral molecules that are specific stereoisomers. And so that's what we can, a part of what we can learn from them. A lot of the interesting areas of research or kind of frontiers of this involve how coordination chemistry is used in enzymes, looking at biomineralization, so the solid state chemistry of inorganic compounds in biomaterials, and then also looking at building modeling systems to study about these different transformations we see in nature, and then trying to learn and glean from them and simplify them. And so that's kind of a broad overview. On the right-hand side, it shows a lot of the inorganic elements that are utilized within our bodies. So charge balance is really dictated by these smaller cations, sodium, potassium, calcium, where a lot of our structures within our body come from calcium and silicon on kind of a macroscopic scale, think bones. Protein structure has a lot of regulation coming from zinc calcium ions, kind of building the protein structure overall. And then nucleic acid structure has a lot more to do with magnesium and manganese. Cell signaling, we see a lot of calcium, NO, and then also zinc involved. And signaling in DNA has a lot of different transition metals involved in that. And this is looking at metal responsive transcriptional regulators. Acid-base catalysis also is going to utilize these metal enzymes as well, looking at zinc, iron, nickel, manganese, and magnesium all playing a role in that. We also see atom and group transfer chemical reactions being mediated by different transition metals. And of course, electron transfer and redox catalysis. It's going to be your body taking advantage of these tunable different oxidation states that are available, and using them to shuttle electrons around. Which is to say, the research possibilities are endless. So let's just take a little snapshot, like a little like, hey, isn't this cool? And let's look at porphyrins, and let's look at the chemistry behind hemes in general. So right here on the left, I've got a porphyrin structure, and this is a ligand. It's a tetradentate ligand. It's going to bind through the lone pairs on these 4 nitrogens that are going to act as your Lewis base. And this porphyrin structure, it basically creates something that happens in the equatorial plane. But do you notice all of these conjugated bonds? Look, there's a conjugated bond system going around this entire gigantic ligand, which means that there are resonance structures here, especially when we remove these hydrogens on our nitrogens and we have some negative charge there. And it also means that even with those hydrogens there, those pi orbitals that those electrons occupy, they're really delocalized in these conjugated bond systems across that whole ligand. And so we have a very smearable, polarizable electron cloud. Which just tells me I can mess with it in some way and tune it, make it do what I want. And so we see this structure in hemoglobin and myoglobin, and I'm going to focus just on these two. So this is a little sliver of bioinorganic chemistry to show some of the interesting things that we take away from what we learned about transition metals. And so this porphyrin structure is incorporated in both of these proteins, and they utilize iron. So the porphyrin structure right here is going to place these nitrogens as the ligands to our iron 2 in the center. And the iron 2 is going to take on an octahedral geometry by coordinating also to a nitrogen on a nearby amino acid from the protein structure. And then that leaves a very key spot open for the binding of oxygen to the metal iron. And so to get that octahedral complex. And when that oxygen's not there, you can envision this is actually a square pyramid arrangement, or possibly puckering into something more like a trigonal bipyramidal arrangement. All right, so looking at iron hemes specifically. And so heme is what we're going to call that porphyrin ligand around an iron. So we've got two different versions we're going to look at. We're going to look at hemoglobin, and we're going to look at myoglobin. And they are found in different parts of the body. Hemoglobin is found in the blood, and its job is to reversibly bind oxygen and transport it throughout the body. And so that means in hemoglobin, that iron needs to form a coordinate covalent bond to the oxygen atom, or the dioxygen molecule to the oxygen atom within it, and then be able to let it go reversibly. So it needs to be easily done both ways. It's easily bind and easily release. Now, in myoglobin, it is a structure that's actually found in muscles, and it's also going to bind oxygen. So its job is to be an iron that sits in this porphyrin, it's connected to this protein, and it also binds oxygen. That's its job. However, its job is to hold on to it a little bit longer, and so it doesn't as easily release it. It's not there to transport that oxygen around. It's there to hold on to oxygen and release it when needed. And so that's a subtle difference, right? Like, how long do you hold on to that oxygen atom? How easy is it to reversibly bind it? It needs to be easy to bind it, period, but how easy is it to remove it? And so let's look at this a bit more. Um, so looking at the heme structure in general, we see we can study the magnetic susceptibility of the actual iron 2 in the porphyrin structure and within the enzyme itself. And we find that the form of iron that doesn't have the oxygen bound to it is paramagnetic. Um, and so we have a square pyramidal arrangement of ligands around this iron, which is going to give us this arrangement of splitting pattern for our crystal field. Just the way those ligands are arranging themselves around those d orbitals. So if we know this is paramagnetic, is this going to be a high spin or low spin complex? If you want to take a pause button, try it out yourself. But now I'll continue. So our iron plus 2 is going to have 6d electrons. It's a d6. And so let's take a look at the difference between the two. Let's start with low spin. So if I'm low spin, I'm going to go 1, 2, 3, 4 before I move up in an energy level. 5, 6. And I know we said for square pyramid and things like this, they're typically low spin. But if this is low spin, it's diamagnetic. Which doesn't match the experimental results for deoxygenated iron heme at all. So let's look at this again. Now let's assume it's a high spin complex. 1, 2, 3, 4, 5, 6. Haha. Now we have a paramagnetic compound. So when it's high spin, it's paramagnetic with 4 unpaired electrons. So that tells us something about the environment that iron's around, that we're not seeing such dramatic splitting of our energy levels that we prevent a high spin complex from forming. And so that gives us some insight into how close those nitrogens are to that iron. It gives us some insight into the different atoms and like the availability of the electrons and the cloud around them as they approach and bind to the metal ion as well. So we can learn quite a bit, especially when we compare it to with the oxygenated form and other forms along there. It gives us insights into the electronic nature of this specific complex, which tells us a lot about the chemistry of hemoglobin. All right, so then we add oxygen, right? And so now magnetic susceptibility, I can't say it today at all. Magnetic susceptibility studies, so are going to be done, and they're going to look at the oxygenated form of our iron heme. And when we do that, we see it's diamagnetic instead of paramagnetic. And so we're now, once it binds that oxygen molecule, it's now a octahedral compound rather than a square pyramid. And so our crystal field splitting is going to change as that oxygen binds. And so let's take a look at what happens when we go low spin. We're still an iron +2, right? And so that means we're still a D6. So if it's low spin, I am going to have no unpaired electrons. So I'm going to have a diamagnetic compound when it's low spin. To show in a different color, if it was high spin, it would be 4 unpaired electrons. So I would expect a paramagnetic for a high spin complex. So that means when we bind that oxygen, we go from a low spin square pyramidal, or sorry, a high spin square pyramidal to a low spin octahedral complex that's diamagnetic. So it's not just a changing of the structure. That structure also comes with a reassessment of whether or not low or high spin is more energetically favorable. But that's influenced quite a bit by the fact that we actually now also have another ligand. It's that oxygen atom. And so it's going to change the very nature of that electronic environment around the iron. Not just the pattern that it splits in, but maybe also how much it splits as well. All right, now that change in paramagnetic versus diamagnetic, the shift in our crystal structure and that splitting energy, it might make it easier or harder for us to be able to bind or not bind our oxygen atom. And I'm going to stop us there before we go. This becomes a 3-day lecture. And now I want to switch gears and look at myoglobin and look at one of the differences between myoglobin and hemoglobin and how it's binding oxygen, because it's doing the same thing. It has a porphyrin structure. It has an iron 2, and it's binding oxygen, but for some reason, it doesn't release it as easily in the same way. What we see is that when our myoglobin, right here's our iron center, binds our oxygen, there is a histidine residue from the protein molecule that isn't binding to our iron at all. It's just hanging out. But what it does is it forms this really critical hydrogen bond. And that hydrogen bond stabilizes that oxygen in a specific orientation. And more importantly, that hydrogen bonding interaction with this oxygen right here allows the electronics of that oxygen to be stabilized to support a negative charge. That negative charge is supported by the attraction to that positive hydrogen, because it's almost like a partial, like, peroxide bond in a sense. And so what happens in the myoglobin is when that oxygen bonds or coordinate covalently bonds to the iron +2, this oxygen right here is hydrogen bonding to this hydrogen on the histidine, which actually stabilizes a redox reaction where we see an electron travel from our iron to the oxygen atom. And so then what we end up with is we end up with our iron oxidized to iron +3 and our oxygen reduced to O2 minus. Again, stabilized by that hydrogen bond to the histidine residue nearby that otherwise isn't bonded to that iron at all. It's just there to interact with that oxygen atom. And so the structure, the 3D structure of this enzyme is changing the way the oxygen atom is actually interacting with that metal ion, such that it promotes a redox reaction that then changes the fundamental bonding structure of that atom. Now we have a larger magnitude of charge difference between the oxygen and the iron. And so that difference in magnitude of charge increases that attraction between our ligand and our metal center of our coordinate covalent compound, right? Because it's that magnitude of charge and distance from Coulomb's law that really dictates how attractive or how repulsive our energy is when we're bringing these charges close together, and we're bringing opposite charges together. We've now increased that quite a bit by making the oxygen molecule negative and increasing the magnitude of positive charge on the iron. And so there'll be a stronger attraction to them and a stronger bond. And that means it's going to be harder to break. And so that means myoglobin is not going to release that oxygen as easily and as readily as hemoglobin will, because hemoglobin doesn't have that stabilizing hydrogen bond to that oxygen molecule. And I just think that's really cool. I just think that's really neat. Um, and that's where I'm going to stop for this lecture series. Your textbook tells you how to prepare transition metals, and I just don't think it's anywhere near as interesting as like the differences between hemoglobin and myoglobin. So I put my time where I think my interest lies and where I think yours should too. And I'll stop there.